Hello everyone, hope you all are doing well. So today we are going to discuss the next answer copy of our Aim for Agriculture series. So as you all know that we are having Aim for Agriculture series in which we are having daily answer writing practice and weekly full tests. And today we are going to discuss the answer copy of full test number 12. So there were 52 daily answer writing practices which we have already finished, and there were 14 full tests, and up to 11 we have finished. We will be doing 12 today, and in 2 days we will completing all the answer copies, and all the PDF of these copies, jitne bhi PDF, all these PDFs have been have been uploaded in the Telegram group, so you can join the Telegram group and get access to all these PDFs, because many of you are commenting and messaging me at different places that you want these PDFs, so you can go and download it there. So let's start. Let's see what were the questions asked and how the student has approached the answer. So the first question was, and I must let you know that I will be a bit faster because it is a full test and there are a lot of questions to be discussed, and if I start discussing line by line, it will take a lot of time. So I will be discussing the important points. I will just giving an overview of the structure of answer, and if you want to read the answers, you can simply pause the video and read it, or you can directly visit the Telegram channel and you can read the PDFs if you want to. So let's start. So the first question is, describe the mode of origin and growth of cell wall with its function. So we all know what cell wall is. It is an organelle that is present in plant cells, while it is an cell wall is absent in animal cell. We all know this normal things. Now we will see its mode of origin. So during cell division, you can see that cell wall forms uh, from the cell plate during cytokinesis. Okay, then there is primary cell wall formation. It is developed during cell expansion, and the primary wall which is formed, it is flexible, it is not that hard. Then the secondary cell wall is formed during a after cell expansion. The secondary cell wall is formed, which is stronger as compared to primary cell wall, and this is kind of on harder side, whereas the primary cell wall is kind of flexible and on softer sides. So this is how cell wall is developed. Now the uh, the second part of the question is asking about growth of cell wall, and you can see that the question is a 10 marker, so the student has not given that much of detail of per part in every part, but it has been well discussed because we have to discuss three parts here: origin, growth and functions. So very well written answer. You can also learn how to address the answers, because many times what happens, many parts are there in 10 markers, so how to address, how to finish those answers in around 150 words. So by reading these answers, you can also get to know how to approach that kind of way. Okay, now growth of cell wall, there is cellulose synthesis is there, then protein and hemicellulose is added, then enzyme activity leads to expansion and loosens the cell wall for growth. Now what are the functions of cell wall? Obviously it provides rigidity to the plant cells, it helps to maintain cell shape, it prevents collapse, it acts as a barrier against pathogens, it controls cell expansion and organ development, facilitates water and nutrient movement. So these are some of the important functions of cell wall, and I think you all know it. Now let's move to the next question. The next question is, what is polyploidy? And we have to give a brief account of classification of polyploids. So polyploidy is a condition where an organism has more than two sets of chromosomes. You can poly, that is more than two. Okay, so it means there are more than two sets of chromosomes common in plant, and it is generally common in plants and vital for crop improvement. For example, wheat. Wheat is a hexaploid. Some varieties of wheats are hexaploid. Now classifications on the basis of origin, we have autopolyploid, we have allopolyploid. On the basis of chromosome sets, we have triploids, diploids, tetraploids, and on the basis of chromosome pairing behavior, we have regular pairing, we have irregular pairing, polyploids. So well written answer and examples have been well mentioned. Now let's move to the next question. The next question is, describe chemically induced male sterility and its importance in crop improvement and seed production. So chemically induced male sterility is simple, in which male sterility is induced by the use of some chemicals. Either it could be some mutagens, okay, either it could be for example ethyl methane sulfonate, it could be some hormones, it could be herbicides. So when chemical are used to induce male sterility, then it is known as chemically induced male sterility, as the name suggests also. Name suggests only. Okay, now what are its importance? Its importance is that it helps in enhance genetic diversity, it facilitates cross pollination, and you can simply pause and read the answers in detail, and examples have been also well mentioned. And the best thing which I liked about examples is that varieties have been mentioned, particular varieties have been mentioned, so that very much enhances your answer. Broad applicability, cost reduction, so 11 points. Now what are the importance of polyploidy in cell seed production, sorry, importance of CIMS, that is chemically induced male sterility in seed production. It helps in efficient hybrid seed production, it increases seed purity, it helps to scale the production, that is huge number of seeds can be produced in less amount of time. So a little answer. Now let's move to the next question. The next question is, what do you understand by vertical and horizontal disease resistance in crop plants? So what vertical resistance is? It is effective against specific races of a pathogens or strains of a pathogens. In vertical resistance, the plant is able to resist a specific race of pathogen. But when you see the horizontal resistance, it provides protection against multiple strains. That is multiple strains. In vertical it is a specific pathogen, but in horizontal to multiple number of pathogens, multiple strains of pathogens. So this is the basic point you should know. Okay, and obviously it is providing against specific, so there will be governed by one or few genes, whereas in horizontal it will be governed by many genes because it is providing for resistance with a broader number of pathogens. Okay, so well 10 points, you can simply pause and read it. Examples have been well mentioned. Now let's move to the next question. The next question is, explain the molecular markers approach in crop improvement. Okay, so as you can see, what student has done, the student has mentioned the molecular marker approach, but it is generally, you can see, its role or importance have been mentioned. Okay, helps in select plant varieties, it offers precise selection, so it is kind of their importance, or you can say role. So here what students should be done, because in the question it is asking that explain the molecular marker approach, so it could be elaborated, it could be broader, made elaborated, perfect examples would be given for molecular markers, so that could be a better structure, and these roles should be explained in a brief. So you can read the answers and you can see where you can improve, because you not need to elaborate these molecular markers approach, because you need to let the examiner know about the concept, because it is explaining the, it is asking to explain the molecular marker approach instead of writing that a role or improvements done by molecular marker or applications. Those things are not asked, it is asking to explain the molecular markers. You can simply pause and read it, you will get to learn, although the importance have been well mentioned, varieties have been also mentioned. Now let's move to the next question. The next question is, describe the method of recurrent selection and write the importance of combining ability in crop improvement. Okay, so recurrent selection is a breeding method aimed at improving the frequency of desired genes in a population by repeated cycle of selection or crossing and evaluation. So this is a fairly defined recurrent selection. Now what are its method? First we will select, then we will intercross, then we will evaluate, then we will again repeat the same process until we get the proper number of plants or proper varieties. Okay, then we will introduce those varieties or new genetic material, whatever you want to say. What are its importance in importance of combining ability in crop improvement? So it helps us to determine the breeding potential of parent line when crossed. It helps us to predict the hybrid performance. It helps us to identify the best crosses for producing superior hybrids. It is crucial for maximizing heterosis. It focuses on compatibility, combinability, breeders efficiencies. All these things can be seen, so you can simply pause and read it. Now let's move to the next question. The next question is, discuss the structure of plant chromosome and its role in genetic diversity. So when you see about plant chromosome, what you can be done here, that a diagram of chromosome could be made highlighting the different parts. You can see in the structure the student has mentioned chromatin, centromere, so these could be highlighted in a simple diagram of chromosome. That could be a better answer, that would have enhanced the marks. Okay, although it is fair, but you can add it here if you have amount of a certain amount of time. So there is chromatin, there is centromere, there is telomere. It is divided into P and QR, that is P is short arm and Q is long arm. Kinetochore is there. So well written points. Now what is the role of plant chromosome in genetic diversity? So it helps in crossing over during meiosis, creates new genetic combinations. It helps in multiple chromosomes, that is polyploidy, and it leads to enhanced genetic diversity. It helps a structured change in chromosome and creates variation. Jumping genes move within the chromosome causing mutations. So these jumping genes are related to mutations. Differences in chromosome number contribute to diversity. Chromosome recombination in hybrid results in superior traits. So well written points, you can simply pause and read it. Now let's move to the next question. The next question is, explain the mechanism of interphase in mitosis cell division. So actually there are two types of phase. We all know that interphase and mitotic phase. So interphase is generally divided into three types, that is G1 phase, S phase, G2 phase. You can see here. This is the mitotic phase, and this proper is the interphase. That is 90% of the time cell remains in the interphase. Now here we have first we have G1 phase, that is here cell grows and produces proteins needed for DNA replication. In S phase, DNA replication occurs, doubling of genetic material occurs. In G2 phase, the cell produces proteins for mitosis, that here the cell is getting ready for mitosis. Then it is prepared for mitosis in G2 phase. The cell is fully prepared for mitosis in G2 phase. So this was about interphase. Now let's move to the next question. The next question is, what is seed treatment, and discuss the role of quality seeds in success of seed production programs. So we all know what seed treatment is, when we treat the seeds applying certain chemical, biological or physical agents so that it improves its performance. There is proper seed protection from fungus, diseases, other things, and overall our yield increases. So we can do chemical treatment, for example thyrum. We can do biological treatment using rhizobium. We can do physical treatment using hot water, cold water treatment. Now what is its role in quality seed production? Obviously it will lead to high yield. It will improve the seed resistance against disease and pests. It will give us better germination. Biofortified seeds provide higher nutrition. So quality seeds lead to uniform crop traits, adding market demands. Obviously the produce will be uniform and it will be highly demand in market, and we will get good prices for this. Suppose there are 6 types of grains, all these are similar, so we will get good prices for this. And there are 6 types of grain, one is good, one is smaller, one is kind of bit of diseased, so we will get low prices. So this is what the student is trying to say. Okay, it obviously increases the quality of seeds. And suppose sometimes the seeds are stored for next year, so from a better quality seeds, the grains will be produced will be better quality, and it will act as a better quality seed for the next year. Okay, so fair written answers, you can simply pause and read it. They multiply faster and support larger production. Now let's move to the next question. The next question is, define aneuploidy and give an account of morphological and cytological functions of aneuploidy and discuss its applications in crop improvement. So what is aneuploidy? So aneuploidy is a condition of having an abnormal number of chromosomes, either through the gain or loss of one or more chromosomes. It arises from errors during cell division. So what are its morphological functions? There could be stunted or irregular growth. There could be chromosome imbalance, which can lead to poor fertility. It can lead to a differ in size and shape. It may have asymmetrical leaves and abnormal flowers. So bulletin points. Now we will see cytological functions. Here the cells struggle with proper chromosome pairing during meiosis. Okay, they exhibit abnormal mitosis, leading to uneven chromosome distribution. It can form univalents or multivalents, leading to irregular chromosome segregation. But there are certain applications of aneuploidy also, such as it helps to locate a specific chromosome, locate genes on a specific chromosome. It allows chromosome transfer from related species for introducing new traits. So new traits can be used. Obviously, it is used in crop improvement. Here we can create polyploid crops with enhanced traits. Traits could be enhanced. Here we can introduce trait diversity, and when there is high trait diversity, the plant is able to adapt in greater, broader environmental conditions, in diversified environmental conditions. Overall, well written points. Now let's move to the next question. The next question is, what is imbibition and difference between osmosis and diffusion? So we all know imbibition. It is a common example there, that when there is a dry log, it absorbs liquid, and we will see that the size increases. So that is simply what imbibition is. That is by which water is absorbed by solid particles of a substance, causing them to swell. So it can occur in seeds, woods, and other porous materials. It is obviously crucial for seed germination, okay, because after water is or moisture is absorbed by the seed, its metabolic activity starts and then it germinates. So you can see a good student tried to explain with the examples also. Now what is the difference between osmosis and diffusion? So you can see that in osmosis, the movement of water is through a semi-permeable membrane from low concentration to high concentration. But in diffusion, it is through molecules is from high to low concentration. Osmosis, diffusion is just kind of opposite of osmosis. So here in osmosis, it requires semi-permeable membrane, but in diffusion there is not necessary requirement of any semi-permeable membrane. Direction of flow, we all know that there is from dilute to concentrated solution, and from here we can see, you can simply pause and read it. Well, little points have been mentioned. You can see drawing have been also made. And the next question is, describe the preventive and curative methods for management of storage pests in pulses. So what are the preventive methods? Obviously we will provide proper storage conditions, we will use clean containers, we will implement physical barriers, routine checks could be done, set traps. So all the things which we need to prevent the pest attack. But in curative, that pest attack has already taken place, we have to control them. So here we can use chemical methods to control, for example use of malathion. We can apply heat to kill, okay, heat treatment. We can store at low temperature, so that freezing temperatures the pest is not able to establish. We can use fumigants. We can introduce natural predators. We can apply other kinds of things too, for example diatomaceous earth to control pests. So these all things can be done. Now give a detailed account of sucrose metabolism in plants. So the synthesis of sucrose, so it is generally takes place in cytoplasm of leaf cells during photosynthesis. So process involved is photosynthesis, and then what happens? There is phosphorylation of glucose. There is formation of UDP glucose. So you can simply pause and read the points. Examples have been well mentioned, and a diagram have been also made how sucrose is synthesized. Now how sucrose is degraded. Degradation of sucrose is generally, you can see it cytoplasm and vacuoles. Hydrolysis is there, phosphorylation is there, fructose metabolism is there, transport of sucrose is mentioned. It is generally transported through phloem, loaded into phloem, then there is phonum transport, then it is unloaded from phloem to the different parts where it is needed. Now let's move to the next question. The next question is, discuss the strength and weakness of public food distribution system in India. So what are its strengths? Obviously it ensures that low income households have access to food. It leads to food security. Essential food grains are affordable at affordable prices. We all know that the rates are rupees 1, 2, 3 for different types of grains. So we all know at present scenario rupees 1 and rupees 2 or rupees 3 are way beyond cheaper. Okay, it creates a stable market for farmers through MSP. The system improves nutritional intake of vulnerable population. We have also TPDS now, that is targeted public distribution system, through which we are also taking care of nutritional intake. It provides essential food during emergencies. So these are some of the strengths. Now what are their weaknesses? There is inefficiencies, a lot of wastage is there. There is problem of identification of beneficiaries. Many times those who are not eligible are also getting PDS, and many times those who are really in need of PDS grains are not getting it. Okay, many times the grain distributed are of poor quality. The long-term reliance on PDS may reduce self-sufficiency, because you are getting grain sets with cheaper prices, so you will not try to work harder to earn more, because you are getting food at very cheap prices. So these are the things. Now what are pesticide formulations, and discuss the different types of formulations used in pesticides. So generally what formulation is, that is in pesticide there is chemical obviously, but with chemical there are different other things also. It could be some powdery form, there could be some suspension liquid added to it, there could be other kind of solutions. So types of pesticide formulations. So by addition we made the pesticides into different things. Either it could be made liquid, either it could be in powdery form, it could be in some solutions. So in liquid you can see it could be in emulsifiable concentrates, it could be suspension solutions. In solid forms it could be granules, powders, beds. In aerosol formulations you can simply spray them in the form of fine mist or fine mist. There could be microencapsulated forms, there could be slow release formulations, there could be biopesticides formulations. Okay, so well written points. So in formulations what happens, the active ingredients are mixed with inert ingredients so that the pesticides efficiency is improved, it is stable at the environmental temperatures environment. So all these things you should know, and you can simply pause and read it. Now let's move to the next question. The next question is, describe the role of brachynosteroids and salicylic acid in salicylic acid hormones in agriculture. So brachynosteroids are used in cell division. They can be helpful in plant enhance plant resilience to environmental stress. They can improve chlorophyll content. They promote flowering and fruit setting. They alleviate oxidative stress by boosting antioxidant enzyme activity. Now, how salicylic acid is helpful? It enhances plant induction of systematic acquired resistance, that is SAR. It regulates stress response. It boosts chlorophyll content, promoting better photosynthesis. It improves root development. It induces cell division. The fair written points have been mentioned. You can simply pause and read it. Now let's move to the next question. The next question is, discuss the iron and protein malnutrition in India, and we should also describe the dietary pattern for mitigating above said malnutrition. So, this in the introduction, as the student have mentioned that there are problems of iron and wide malnutrition is there, but here we should try to add some kind of example data. For example, you can see the National Family Health Survey 5 has been mentioned here. These examples can be also mentioned in introduction, that would enhance your introduction. Okay. So first we will discuss iron malnutrition, which is the cause of anemia. So we all know that over 50% of women in India, around 50% of women in India is anemic due to this iron malnutrition. They reach in inadequate intake of iron-rich foods. Obviously, it leads to fatigue, impaired cognitive functions, reduced work capacity. Now, about protein malnutrition have been also mentioned. Many children are suffering from kwashiorkor or another protein diseases, marasmus, kwashiorkor. Around 35.5% under 5 are stunted. So, written points. Okay, stunted leads to stunted growth, muscle wasting, weakened immunity. It leads to kwashiorkor and marasmus, which just I discussed. Now, what could be the dietary pattern? We can increase the intake of iron-rich foods such as spinach, legumes, fortified cereals could be introduced. A balanced diet should be maintained using vegetarian or non-vegetarian things. If someone is vegetarian, the vegetarian sources of iron and protein should be introduced to them. Rich foods such as vitamin C and other things would be introduced. Fortified should be consumed. So similar points and very nicely written points. You can simply pause and write. Government initiatives such as Poshan Abhiyan could be helpful. Okay, so now let's move to the next question. The next question is, enlist the seed species grown in India. Briefly discuss the cultivation of post-harvest management practices for coriander cumin. So there are various seed species. Obviously, uh, for all crops we have seed species which we grow in India. Okay. So what happens when we are writing the crops? You can simply add a variety of seed which would be more beneficial. So you can simply pause and write, pause and read. Now we have to discuss the cultivation of coriander and cumin. So coriander, it requires well-drained loamy soil. Obviously, climate have been mentioned, sowing have been mentioned, irrigation have been mentioned, post-harvest management have been mentioned. What is it that directly has been questioned? Oh, actually, it discussed the cultivation and post-harvest management both. So a student has written in brief because uh, the question is 20 marker of course, but there are too many things to address in the questions. So a brief answer is well, and I think well written answer. You can simply pause and read it. Now let's move to the next question. The next question is, Discuss the integrated management of nematodes in fruits and vegetables. So obviously in integrated management we will use cultural practices, we can use resistant varieties. You can simply pause and read it. For cultural practices you can use crop rotation, sanitation, soil solarization. Various kinds of resistant varieties can be used. For example, Pusa ruby has been written. It is a tomato variety. Biological control can be done, okay, by using the organisms. Adult could be predator or other competitive organisms. Chemical control can be done. Organic amendments can be done. Soil health could be enhanced. So this is how through which we can integratedly manage nematodes. So this was the answer, and I hope you pause the video at several intervals and read the answers. And the PDFs, as I have discussed in the start of the video, also that PDFs have been uploaded in the Telegram group. So you can simply join the group and get access to these PDFs. So that is all for today. If you liked it, please press the like button. Please do share and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis and bring such courses for you and in regular intervals. So if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.